as far as I can see, I see 4 people online. Just send me a message, make sure everything's hear me okay and everything's running reasonably okay. All right, so last thing that I did, or what we talked about on Monday, or what I talked about on Monday, was largely about using Bowtie, or sorry, using RSEM as a mapper. So RSEM calculates expression, and it does so by using a mapper to map reads to these genes, and then basically counting how many reads actually map to each individual gene. So if I'm going to look at that output file, all right, so everything that I made from RSEM is in this expression directory. So in this directory, I have my output from RSEM, which is a genes result, an isoform result. So, oh man, whoops, something has gone wrong. Okay. So if I'm looking at this file before classes, I'm messing up. I'm doing stupid things. So what I wanted to spend more time on today is what I'm really talking about today isn't a whole lot of Linuxy things or running programs or anything like that. Today is just kind of looking at output. And so one of these files that I feel like I need to talk about are these BAM files. So these BAM files are effectively they are a file format that's ubiquitous with genomics. It's as important as a FASTA file or FASTQ file. These things are everywhere and they store information. And what these SAM and BAM files are, these are alignment files. These are the files that describe which read map to which gene, and things associated with that. So we can't zcat a BAM file. We can't cat a BAM file. It's in this binary, and this binary can only be read and interpreted and opened with a program called SAMtools. So SAMtools is awesome because it's the tool that helps us read these BAM files or these SAM files. So BAM and SAM are the same thing. It's just BAM is in binary and SAM is in plain text, but they both hold the same amount of information. So SAMtools has a lot of different tools and a lot of utility associated with it. Probably one of the more common, more frequently used tools is this SAMtools view. And so this SAMtools view lets us look and convert between SAM and BAM formats. So if I want to actually look at this file content, I would have to do a SAMtools view on my sorted BAM file. I want to start with the sorted BAM file first. I'm just going to head this first. I'm going to head it because this whole file, right, is 2.3 gigabytes big. It's effectively the same size as our FASTQ files combined. But let me actually look at this for a second. And so what this file holds again is the mapping contents. So it's going to tell us it's going to give us this first column is going to have a read number. There's this flag that says how this read mapped. I mean, like, did it map with its pair? Did it not map with its pair? Is it in the forward orientation? Is it reverse orientation? But these numbers tell you kind of what happened. Then it tells us what, in this specific instance, it tells us which gene it mapped to, which position in this gene it mapped to, a score. This is a map score of how well this read mapped. It's also kind of a uniqueness score of measuring how many places this could have mapped in a genome or in our reference. A little bit of information, and then after that, it's a little bit of information about how this read mapped, and then the sequence of the read itself, and then the quality of the sequence. So both of these are found in our FASTQs. So all this stuff wraps around. I'm going to do some magic to get one line of a SAM file to go across multiple lines. So again, we have the read name, how it mapped, forward, reverse, which gene it mapped to, where in the gene it mapped to, how well did it map, some information about how it mapped, additional stuff, the sequence, and the quality. And that's kind of all we need to care about in a SAM file. And so in our sorted SAM files, what we've done is what initially happens. So what SAMTool does is that it takes the first read in our FASTQ files and say where did this go, and then it's going to take the second read and ask where did it go. And so that information is initially stored in the transcript.bam. So I'm just going to look at the first read in the raw BAM file. And so it's going to tell us this SRR, this read name, and it was mapped first. A dollar says that this read is the first in our FASTQ sequence. And so I'm going to check that real quick while I can. Clean one, head, whoop, I didn't want to do that. Let's see. Let's see. Yeah. So that first read that popped up in the BAM file is the first read that we would find in the FASTQ file. And so what we do whenever we sort, and SAMTools is responsible for sorting. And believe it or not, sorting is a very important algorithm, not only in computer science, but also bioinformatics. So sorting is its own, in computer science courses, sorting is one of the first algorithms that's really described in depth. And there are several different ways to sort something out that we don't need to worry about. But sorting is always important because how we sort our BAM file is not based off of what read went where, but it's looking within a gene and mapping out and telling us which reads map to gene 1 position 1, which reads map to gene 1 position 2, etc., etc., etc. So it gives us a coordinate output. And so it's much easier to look through a sorted file than it is to look for an unsorted file. In a lot of cases, in tools that use BAM files downstream, a lot of them require a sorted BAM file or else nothing will work. And so in our raw BAM file, and I saved this for a little bit, so I have this sambottom.sam file that I made. And so we do keep track of every read in a FASTQ file. It may be flagged as mapped, or it may be flagged as unmapped. So everything at the bottom of a sorted file is unmapped. So these are reads. that were unmapped. They were in the FASTQ file, but they didn't seem to be derived from any of the sequences in our reference CDS file. And so we see flags that basically tell us this is unmapped. So this flag of 77 means it's unmapped, but it's not or anything like that. We just see a bunch of stars and zeros that basically said that this read was unmapped. No, not entirely surprising. But what's nice about SAM tools is that if we want to get rid, we can get rid of those. So we can set a flag in our SAM tools view that we don't have unmapped reads on our BAM files. And the advantage of that is to keep our BAM a little bit smaller, right? More or less save space. So another nice tool of SAM tools is Flagstat. So Flagstat, we have all of these flags that tell us if a read is paired or unpaired, if it's mapped. Did it map with its pair? Is it in the forward orientation? Is it in the reverse orientation? Right, that's stored by this number, our second number. This is our mapping flag. And so what SAM Tools Flagstat does is that it reads through the entire BAM file and it tells us how many reads mapped, how many didn't reads map, and some other other flags that tell us how reads map. So if I do this SAM Tools Flagstat, um, it's going to take a while to read through this entire file. It will take a couple of minutes. Again, the whole reason I'm a little frustrated, so we really won't see a BAM file ever again for the rest of the semester, even though this is probably one of the more important files in genomics. It's just we kind of are running out of time, and honestly, today is probably the last day that we're going to spend on Linux. After this, we're going to spend a lot more time in R. So I uploaded a assignment that basically says the second part of it is to download R if you don't already have it, and install RStudio. And so we'll explore what R and RStudio for the rest of the semester. We really won't be on the server that much from here on. All right, so this is done. And so what this tells us is saying this is the number of reads that were in our FASTQs. This is the number of reads that were attempted to map. So it's about 57 million paired reads that were in our FASTQ file. And out of those 57 million reads, 44 million of them were mapped to our reference sequences. And so that's about 78% of our reads were derived from these protein coding transcripts. The other 22% are either from non-coding RNAs or they're from unannotated regions of the genome. They could be from transposable elements. Who knows? But nonetheless, so what RSEM does is by default, bowtie, it will try to map everything. So it's going to try to map every read, and even if say the left-hand read maps to the genome, but the right-hand read doesn't map to the genome, it's still going to report that. It's still going to hang on to that information. But what RSEM does is that it sorts the BAM file before we do anything with it. So it's already going to get rid of mates or reads that where one read maps to the gene, but the other read didn't map, or say one read mapped to gene A, but the second pair mapped to gene B, something wonky like that. So it's just going to get rid of all that stuff and just keep the reads that mapped and paired appropriately. So that's really all that we see. We don't see anything else about singletons or pairs that map to different genes or things like that. So RSEM just sets SAM tools to get rid of all of that stuff. So all that we have are the paired reads and how many of those paired reads mapped appropriately. And here it's about 78%. which is pretty nice. I have definitely worked with files, some BAM outputs that's like between 40 and 50%. In those cases, for kind of already have similar genomes, um, so you tend to see a lot of reads mapped to mice and humans and other model organisms. All right, so what else is our gene results? So the BAM files are produced by Bowtie. They're produced by any mapper, um, BWA, Star, Bowtie2, whatever, is produced by the mapper. And just kind of as a segue, so these BAM files, this SAM tools, um, BWA, and another tool called BCF tools, they were all derived and designed by one person. So kind of the Gandalf of bioinformaticians is Hang Lee. He was a research scientist, I think at MIT, at least for the Broad Institute, that does a lot of genomic sequencing. He worked in specifically a human evolution, human population genetics lab, and develop these tools to make life easier for pretty much every other person that wants to do bioinformatics. So he himself made BWA, which is one of the more popular mappers. He himself invented the SAM/BAM format around 2009 that everybody uses. So in a lot of ways, single-handedly, he made the fundamental tools that we use in bioinformatics, tools that I couldn't even read. So nonetheless, I think that's just kind of impressive that a lot of these tools are derived from one person around 2007, 2008, plus or minus. He's gone on to be a PI and whatever, but that's just kind of a segue. But moving on is to actually look at the output of RSEM. And so the output of RSEM is actually counting the number of reads that map to each genome. So before I talk about that, I guess one last tool of SAM tools is going to be this depth option. And so what this depth option means is for any given base, how many reads map to that specific base. So if I do a SAM tool depth on deliver transcripts for to BAM. All right, so what this is looking at, so at this gene, GNAI3, at base pair 2, there were 2 reads that mapped to this base. At base pair 10, there were 5 reads. At base pair 22, there were 6 reads, et cetera, et cetera, et cetera. Throughout this entire sequence, it's just telling us for every single base how many reads overlap that specific position. So I think in this gene, it maxes out at about 150, 106 reads that map to a specific base. So this base pair 222 has about 157 reads mapped to it. And so usually you see this coverage of a gene is not universal. Even though that's what we should expect, um, that's not always what happens. So we tend to get an average depth of every single um, of every gene. Right. So yeah, it's definitely not homogeneous. So we were at 150. Now at base pair 700, we're at 53 reads, and then at base pair 800, we're at 170 reads that map to those positions. So definitely not always homogeneous. Um, but anyways, back to the output of RSEM. So the gene results and the isoform results are basically the same file. I don't ever look at the isoform results. It doesn't tell us that much more information than what's in the gene result file. And it is a plain text, so we can look at this. And so what we see in this file is basically gene expression data. So one, it tells us this gene name, and it kind of repeats the gene name for transcript name, whatever. But then it tells us this length of the gene, the effective length of the gene, and how many reads map to it, and then these expression metrics that normalize the data in some way. So if we have FASTQ files that say have 2 million reads in them. And then we have a FASTQ file from the same tissue, but it's going to have 100 million, or sorry, 10 million reads in it. So we have one that has 2 million and then another that has 100 or 10 million. Just by default, more reads are going to map to our references from our 10 million read file versus our 2 million read file. So we need to account for that variation in sequencing depth. So this raw number, so this expected count, this is the fifth column. 1, 2, 3, 1, 1, 2, 3, 4, 5. This is our fifth column in our RSEM output file. So that's 1, 2, 3, 4, 5. So this 58 number, that does tell us about 58 reads mapped to our GNA3 gene. That is our raw count. This conversion, this TPM number and FPKM number are kind of similar, or they're related. So what FPKM stands for is fragments per kilobase million. And so we normalize our data based off of the size of the library that we mapped and the length of the gene. So when we say fragments per kilobase million, it's saying how many fragments map to this gene, and this gene is say 3,000 bases. So our kilobase is the length of the gene, how long the gene is in the number of thousands of bases. So if we have a 3,000 base pair gene, the K is, we denote that by 3. We reduced by 1,000, so that would be 3. And then the million per million is the million number of reads. So if we have 2 million reads that mapped, what that's saying is we may have 58 reads that map to a 3 kilobase gene, but we're also going to divide by the number of reads in terms of millions. So if we have this is poorly explained. So we take the number of fragments that mapped, we divide it by the gene length in terms of kilobases, and then we divide that number again by the number of reads that mapped per million. So it normalizes based off the length of the gene, and it also normalizes based off the length of the library that we map to. And so this makes doing an RPKM, doing this normalization, allows us to directly compare, or somewhat directly compare, reads that a library of 2 million versus a library of 10 million, right? We're just kind of taking percentages in some degree from there. And this TPM is also further normalized. And so what it's doing is that if we sum up all of the FPKM numbers, so 45 plus 0 plus 0 plus 1, 1.3 plus all of these numbers to get our total. And then we take this number, 45, and divide by the total number of reads, total number of RPKMs. That's going to give us a percentage of RNA that was present in our pool. And so that's what our TPM is. It stands for transcripts per million, but it's still effectively a percentage multiplied by a million. So 45 divided by the total RPKM will give us a very small number, like 0.00036, if not smaller. And so when we multiply by a million, what that basically says is that out of a million reads, or out of a million transcripts that we're going to find in this cell, 36 of them are derived from this gene. So it's kind of low. It's not low. We have 20,000 genes, several thousand transcripts. So it's going to happen. We're going to see a lot of genes with expression, these TPMs that make up a very small percentage of the total transcriptome. But that's what this TPM number is effectively trying to be, is that it's a percentage of the total. but it's a total out of a million reads. So if you do sum up the whole TPM column, it's going to total 1 million. Right. Um, so if we were trying to manipulate this file, or at least extract some information out of it without actually looking at it, we can again use our cut and sort tool. So if I want to look at the number of reads that map to each gene, I may cut out the fifth column of our gene results. Okay, right. So this, I believe, this is the number of reads. 285, 258, 00, 13. Yeah. Okay. So this is the number of reads that map to each individual gene. And so I might want to know how many genes did not have any expression. So I made grep specifically on 0.00 just to make sure. Yeah. So let's see. Yeah, it's gonna be close enough. So I may count how many lines had the 0.0 in it, and so that's about 41,000 lines. Um, that's not right. I'm doing something silly. Because they're already going to have, let's see, how many lines are actually in this file. Gene, 68,000 lines. So I've done something wrong. I promise, like 41,000 are not unexpressed. I'm pretty confident that's not right. What's happening is that if I grep on the 0.00, I'm not just getting the zeros, right? So there's one with 20.00. So this is just something stupid that's happening. There's 70.00, whatever. But let's see. But I can find out what's my most expressed gene, or at least which gene had the most reads mapped to it. So I can still cut on that fifth column, sort it as a number, and then I can get all of my read counts per gene. And so this is my biggest one. That's my biggest number by far. So I may want to grep this number out. And so it's going to tell us this ALB gene had the most number of reads mapped to it, right? So this may not necessarily be my most expressed gene, because it could just be really long. So I inherently have a lot of reads mapped to it, but it does, nonetheless, this gene had the most number of reads mapped to it. If I sort on the TPM column, I'll probably get a different gene being expressed as considering the most expressed gene, when we take into consideration the number of reads that we mapped and the link of our gene. So if I do the same thing on the sixth column, this is our TPM column. So I made grep on this. Right, so now it's saying a mitochondrial ATP8 gene is the most expressed gene. Not that surprising. So depending on which metric we look at, number of raw reads or corrected number, a corrected expression value will tell us something a little bit different. But we're going to revisit. So you definitely need to do this. So you need to get this gene result file from your FASTQ files. You need to get that generated because we will revisit this stuff to look for genes that are differentially expressed in the kidney versus the liver. But first, I mean, the primary tool that does that is R. And we need to get a little bit more comfortable with R before we do a differential gene expression. So we at least need a couple of weeks of practice, 2 or 3 weeks of practice with R before we do a differential gene expression analysis, but we do need to generate this first, and then we'll worry about that later. All right, so that's all I really wanted to talk about today is just the BAM files, and also the gene results files, the output of RSEM. And yeah, we'll go from there. That's at least it for today. That's all I want to talk about, I think. If not, I'll send out an email or something else or do another video, who knows. But I think that's all I wanted to talk about to now. So I'm gonna kill the stream now.